Okay, can you see now? Just one of you can unmute and read the page if you can see.
Yes.
Excretion and osmoregulation.
Excretion and osmoregulation. Yes. So we are starting with this chapter, excretion and osmoregulation. We'll first try to understand what does it mean. What does excretion mean? So excretion is first step in that is the separation of metabolic waste products, mainly nitrogenous waste products, then collection of this and then elimination of this. So excretion is the elimination of nitrogenous and other waste materials from the body. Now whatever the waste is produced in our body, that is thrown out, that is eliminated. And for that, first it should be separated, all this metabolic waste, and generally it is in the form of nitrogenous compounds, nitrogenous forms, so nitrogenous waste, and then it is collected and then eliminated out of the body, thrown out of the body. So that is what excretion. Okay. And in case of human beings, this what is the excretory system includes, and what are the organs involved in the system. So what are the parts in the system involved to excrete all these nitrogenous waste that we are going to study. So these are sent out, these nitrogenous wastes are sent out along with some water, and that is what excretion. Along with that, these kidneys, which is considered the most important part of the excretory system, these are very much involved in the homeostasis of the body fluid. Homeostasis is the process where the internal environment of our body is maintained, where salt and water balance is made. And water is the most important constituent of the protoplasm, that is the living substance which is present in all the type of cells. So this is what the structure of kidneys, where kidneys also maintain acid base balance by excreting hydrogen ions, H plus ions. So not only excretion but maintaining the internal environment by homeostasis, as well as maintaining the acid base balance, maintaining the acid base balance by excreting hydrogen ions, that is pH is maintained by kidneys. Now there are different modes of excretion. What are these? These are the three main types of modes of excretion: ammonotelism, ureotelism, and uricotelism. So based on the nitrogenous waste, modes of excretion are of three types. First, when the excretion is in the form of ammonia, that mode of excretion is called as ammonotelism. When the excretion is in the form of urea, that is called as ureotelism. And when excretion is in the form of uric acid, that is called as uricotelism. Animals cannot eliminate free nitrogen, but can eliminate it in the form of nitrogenous end products. And the metabolism of excess amino acids, nitrogenous basis of nucleic acids, namely mainly pyramids, these nucleic acids which include pyrimidines, they produce ammonia, from which urea or uric acid is formed depending on the need. Everybody knows that whatever the food material we take in, that contains most important main component in the form of nitrogenous compounds, N, P, K. These are the main nutrients. These nutrients in the form of this, what it contains, NPK is major factors. That means N is what? Nitrogen. So most of the compounds which we are eating, these show the presence of nitrogen. And what are these products from which contain amino acids, then nitrogenous basis of nucleic acids? If you remember the structure of nucleic acids, what these nucleotides contain, that is nitrogen bases like pyrimidines and pyrimidines, purines and pyrimidines. Then pyrimidines, they are of two types, purines are of two types. What all those contain, the main thing is of nitrogen, right? So along with that, some other compounds also present. So these are excreted. Excess of these or different forms, they are removed out of the body when they are in excess, when they are not required. Okay. Then first mode, that is ammonotelism, that we will discuss in detail now. As I told you, elimination of ammonia as the chief nitrogenous waste material is termed as ammonotelism. Ammonotelism means when the excretion is in the form of ammonia, that is formed by oxidative deamination of amino acids. That is the process. Deamination, that is the reverse process. Amination, amino acids are produced, but from amino acids, different forms like ammonia, they are produced. That is deamination. So deamination chiefly occurs in the liver of vertebrate body. And the amino group separated from the amino acid combines with hydrogen and becomes ammonia. So from amino acid, amino group is separated, and when it combines with hydrogen, it forms ammonia. So this is ammonotelism, where it takes place in the form of ammonia excretion. So excretion of ammonia is most common in aquatic species. So many of the lower invertebrates expel this ammonia through whole body surface by simple diffusion. Now everybody knows what diffusion means. You have learned it. Diffusion is what? When there is movement of the particles or the substances or the molecules from the higher concentration to the lower concentration, that is generally takes place by the diffusion process. So in most of the lower invertebrates, this type of simple diffusion is used to expel ammonia from their body. But in bony fishes, most of the ammonia is lost by ammonium ions across the epithelium of gills. With kidneys, excretion only minor amount of nitrogenous waste. Means most of the excretion in case of bony fishes takes place through gills. So this is ammonia is lost. That is in the form of, that is considered to be nitrogenous waste. So the animals which excrete ammonia are called ammonotelic animals. Remember, ammonotelism and ammonotelic animals. And the most important example, or the example of ammonotelism or ammonotelic animals, is bony fishes. Most of the aquatic invertebrates, they show such type of ammonotelism. Bony fishes is the best example you can remember of ammonotelism. Now the next is ureotelism. Ureotelism means what? When the excretion is in the form of urea, then that is called as ureotelism. Terrestrial adaptations, generally they produce lesser toxic nitrogenous waste such as urea and uric acid for the conservation of body. Now, you know, very simple to remember in case of ammonotelism and ureotelism. Ammonotelism, where ammonia is the excretory product, ammonia is very toxic, highly toxic, which requires lot of water, because this ammonia is released in the water, and then only that, because for this, whatever the ammonia is released, the amount of water is required is very much, and that's why only aquatic animals, in case of aquatic animals, that too bony fishes, they show ammonotelism. But terrestrial animals, where they have, compared to aquatic animals, less availability of water, so they have adapted in such a way that they can release such a nitrogenous waste which can be dissolved in less amount of water also. So terrestrial adaptation, this is one of the examples of terrestrial adaptations, where they produce lesser toxic nitrogenous waste like urea or uric acid, and so that they can conserve water in their body. So it is produced in vertebrate liver by a metabolic cycle, that is ornithine cycle. This is called as ornithine cycle, that combines ammonia with carbon dioxide. Now, if you we discuss whatever NH3 is produced, or ammonia is produced, that is really which combines with hydrogen, and then ammonia is produced. NH3 combines with hydrogen, and then NH4 is produced. But here what it is done, that metabolic waste, whatever, combines with carbon dioxide, right? That combines ammonia with carbon dioxide in case of ureotelism. And urea is 100,000 times less toxic than ammonia. As I was telling you just now, that ammonia is highly toxic, and For that, the amount of water is needed in a very large quantity. So only aquatic animals can show a monotelism, whereas ureotelism, that urea being less toxic, almost 100,000 times less toxic than ammonia, this type of phenomena, excretion of urea in the form of urea when excretion takes place, that is ureotelism, shown by the animals, that is terrestrial animals, and where they transport these animals, these permits these animals transport and store urea safely at higher concentrations. That means to some extent they can store urea in their body and then it is mixed with water and released out of the body. So circulatory system transports this urea for filtration and then elimination. That is for ureotelism. Thus, urea excreting animals require much lesser water and some amount of urea may be retained in medullary fluid of kidneys to maintain the desired osmolarity. Okay, so this is what just ureotelism you remember. So examples of ureotelism: earthworms, cartilaginous fishes, most of the amphibians which spend most of the time on land. You know what is the meaning of amphibians, which can live in water as well as land, but most of the period of their life cycles they spend on land. So these amphibians also show ureotelism. And mammals excrete urea and their chief nitrogenous base is urea, and so they are all called as ureotelic animals. So remember that ureotelism, which show which where the urea is excretory, their excretory product. Now the third type of mode of excretion is uricotelism, where elimination of urea is in the form elimination of just a minute, I will just okay. So uricotelism is elimination of uric acid. That means uric acid is the nitrogenous waste which is considered to be the nitrogenous waste, and when it is eliminated, nitrogenous waste is eliminated in the form of uric acid. That type of excretory mode or mode of excretion is called as uricotelism. So uric acid is mainly formed from ammonia, mostly in the liver of liver as well as Malpighian tubules of tracheate arthropods. You just remember the arthropods as well as avians, that means birds, those all excrete their waste in the form of uric acid. So uric acid is less toxic than urea. So remember, ammonia is highly toxic, urea is moderately toxic, then uric acid is again less toxic than urea, and being insoluble in water can be excreted as semi-solid pest or pellets with very little water loss. I'll just give you here one example. If you have seen the waste of mice or mouse or rats, then you must have observed that they excrete, that their excretion is in the form of small dry pellets, isn't it? So that is what uricotelism. This is a great advantage for animals with little access of water, where they show, means wherever there is very less availability of water in such areas, especially which are living in the desert areas. So most of the reptiles, then birds, they excrete their waste in the form of uric acid, which is the major nitrogenous waste, and all these are called as uricotelic animals, and the mode of excretion is uricotelism. Now this is the table which explains that a monotelic excretion is ammonia. Ureotelic excretion is in the form of urea. Uricotelic excretion is in the form of uric acid. And the animals which excrete ammonia, they are most of the aquatic animals, for example, bony fishes and lower invertebrates. Whereas ureotelism is seen in earthworms, cartilaginous fishes, amphibians, and mammals. You can remember ammonia excretion is seen in bony fishes and lower invertebrates, whereas cartilaginous fishes show ureotelism. And uricotelic animals, their example is arthropods, reptiles, aves, and land snails, where they excrete uric acid. When toxicity is considered, ammonia is highly toxic. Urea compared to ammonia is low toxic, whereas uric acid is lower than urea. That is less toxic than urea. Okay. Well, amount of water required for excretion in case of ammonotelic organisms, it is high. Large amount of water is required. For urea, moderate. You remember the example of ourselves, human beings, urea. Here, aquatic bony fishes, ammonia, and here uric acid, where very less amount of water is required. Reptiles, all the reptiles, you can remember the examples, rats, etc. Isn't it? Then energy required for the production of ammonia is high, less, very less. Then for urea, it is moderate, and for the uric acid, it is very high energy is required for its production. So this table explains everything about the modes of excretion. So organs responsible for the elimination of these metabolic waste products, they are different in different animals. Their system, excretory systems or excretory organs present in different animals, that differs from animals to animals, where in case of invertebrates, the structures of excretion are simple, whereas in case of vertebrates, these structures are complex. Now we will talk about the human excretory system. What it includes. Remember, number one, pair of kidneys. Number two, pair of ureters. Number three, urinary bladder. And the number four, that is urethra. I will repeat again. Pair of kidneys, pair of ureters, this tubular part. Then this pouch-like structure, that is urinary bladder. And the small tubular structure, that is the urethra. So these four parts are involved in the excretory system of humans: kidneys, ureter, urinary bladder, and urethra. We'll discuss all these parts stepwise in a stepwise manner. A pair of kidneys, this is one, and this is the second. So this is what pair of kidneys. These are the chief excretory organs in man or human beings, you can say. What is the color? It is dark red, and the shape is bean shaped, and it shows, which is covered anteriorly by adrenal glands. So adrenal glands are present here on the anterior part of the kidneys. Now if you see the position, they are protected by last two pairs of ribs, which are 11th and 12th pairs. If you see, this is the rib cage, and the total 12 ribs, if you know, everybody knows, if remember, 12 pairs are present. 11th and 12th pair, here it is located, that extends from 12th thoracic vertebra till the third lumbar vertebra. So this is the position of kidney in, that is the location of the kidneys. So kidneys are present on either side of vertebral column, either side of vertebral column. These are covered by parietal peritoneum only on their ventral side. Ventral side is what? The front side. Dorsal side is back side. So on the front side only, the parietal peritoneum is present. That is, kidneys are covered by parietal peritoneum only on their ventral side. One more thing, you can see the position. One is little bit in the lower position and the other is seen little bit on the higher level. You know why? The reason is like this. Because of the huge liver present over here in this region, this right kidney is lower than the left kidney. You remember, this is left kidney and this is the right kidney. Why this is placed little in the lower position? Because of the this area which is present above this right kidney, liver is present, and because of that this is little bit low in the lower side. Now, what is the length of each kidney? Near about 10 centimeter. Width of each kidney is about 5 centimeter, and the thickness is 4 centimeter. So these are the measurements. And if you see the location, as I told you here, 11th, 12th vertebra, that is thoracic vertebra, and then it is continued till third lumbar. That means it is present between the thoracic and abdominal cavity. So this is the location of kidneys. Then weight, if you see, the average weight of kidney in man is 120 to 170 grams. That is the average weight. In males, it is near about 150 grams. Now, when you study the internal structure of kidney, it looks like this. Means that is the longitudinal section of kidney. When you take the longitudinal section, that is LS, longitudinal section, or we can say VS, vertical section of kidney, how it looks. See, this is the outermost layer, that is called as cortex, and this is the middle layer, middle part, that is called as medulla, and this is the innermost part, that is called as pelvis region. Okay, so this is pelvis, this is medulla, and the outermost part, that is the protective, that is the cortex. Okay, the inner concave surface has a deep notch called hilum. So whatever the notch here, you can see this deep notch, that is called as hilum. And what is the role of this hilum? That at the point which the where at which the renal artery and nerves enter, and the renal vein and ureter leaves. See, this is the renal artery, it is entering, nerves are entering, and this is the renal vein, and this is the ureter which is leaving. So this notch is called as hilum. Okay. So what are the parts? Remember, when you take the longitudinal section, the outermost cortex, middle one medulla, and the innermost, that is the pelvis, which shows a notch here in this region. Then through this notch, the renal arteries and nerves enter, and renal vein and uh, ureter leave. So this is the structure. Now again, you can remember here, the longitudinal section of kidney. This is again in the another diagram. You can see this part where I am showing you with the help of arrow. This is the cortical region, or this is also called as cortex. And whatever the pyramidal structures you can see, this is the medullary part or medullary region, and the most here funnel shaped structure, this is called as pelvis, innermost region. So how this kidney is divided? First, outermost is cortex, middle is medulla, in the innermost region, this is called as pelvis region. Okay, from where that enters, this calycis enters this ureter. Okay, so this is the ureter, this is the hilum. Now, the next diagram, just remember, as I told you, all the same things, these pyramidal parts in your textbook also, you keep your textbook open and see the page number 15.5, that is LS of kidney, where all different parts are shown. You keep this diagram ready with you so that you will understand. Again, I'll repeat the parts, three regions, that is outermost cortex, that is the cortical region, the middle where you can see all these pyramids, that is the pyramidal region where you can see it is shown here in the diagram, that is what called as the medullary region, and the innermost, that is pelvis region. Okay, now these pyramids are called as renal pyramids, or they are also known as renal papillae of pyramids. And you can see how they are arranged. This is what called as the base. Okay, here this pyramidal region, if you see, this is uh, this pyramidal region that is having, this is the base and this is the apex. Means apex is what, somewhat narrower, which is towards the pelvic region, pelvis region, and this base is towards the cortical region, which is broader. So this is the broad region called as base of the pyramid, and this is the apex of the pyramid, which is little bit narrow, which is towards the medullary region. Okay, and then in between the previous diagram, if you see, you can see this diagram here, this in between these pyramids, can you see these columnar structures, these columns, these are called as columns of Bertini. So these columns are called as, in between the pyramidal region, the columns are present, and these are called as columns of Bertini. So this is the internal structure, you remember, where you have to remember all these points, that is cortex, medulla, pelvis, then cortical region, inside the cortex whatever is present, that is in between, in the medullary region and the cortical region, this pyramidal part which is present, that is generally present in the medullary region, these are called as the renal papillae of pyramids. Then the next, that is the columns which you can see, they are also called as the columns of Bertini, or they are also known as renal columns. So the names are one and the same. These are called as renal pyramids, and in between the pyramidal region, whatever the columnar structures you can see, these are the renal columns. So this is the internal structure you need to remember. The columns of Bertini or renal columns, one and the same, which are represented here in this diagram by these arrows. Arrows whichever are shown here, this showed by the structure like this, rounded structures, rounded parts. Okay, this is what you have to remember. Then one more point you need to remember is the process of passing out urine is called as micturition. That is a small part, box given in the textbook that you read in detail. So how it takes place, how is the working of this micturition. Okay, now the next part of the excretory system is ureter. Now we studied about the kidneys, pair of kidneys, its length, location, width, thickness, and even internal parts. Internal part, that means LS of kidney also we discussed just now. Now the next part included in this is, that is a pair of ureter, which what you can see, this tubular structure is called as ureter, which is starting with this pelvis region, which is starting from the kidneys, and that joins these kidneys to urate, what is that called as urinary bladder. So this tubular structure, what is the role of this tubular structure called as ureter, which is receiving the urine, that is the nitrogenous or waste product, that is in the form of urine from the kidney, and it is taken to the urinary bladder for the storage. So you can remember in this way also, kidney where there is formation of urine, and through the ureter it is transported. So synthesis of formation in the kidneys, ureter for the transportation, urinary bladder for the temporary storage, and urethra for its excretion. So in this way also, in one word point, you can remember the functions of each part. Now ureter is nothing but a pair of slender whitish tubes about 40 centimeters long. And what it carries? It carries urine. From where? This part which emerges from the pelvis of kidneys. This is what pelvis of kidney. And where it is taken, it is transmitted or transported from these tubular structures to the urinary bladder. And the wall of this ureter, they are lined by transitional epithelium. Now the next structure or the part involved in this excretory system is urinary bladder, where there is temporary storage of urine. So it is a median storage sac. situated in the lower abdominal cavity. This is a pouch-like structure or sac-like structure which is situated in the lower abdominal cavity. So it looks like this. This is muscular, thick, with the distensible walls, and it's lined by transitional epithelium. So this is the structure of urinary bladder. It stores urine temporarily. Generally 500 ml to 1 liter of urine is temporarily stored in the urinary bladder. Now the next and the last part involved in the excretory system of human being is this is small tubular structure called as urethra. This is the structure where this is external, where it shows external urethral sphincter muscles which are made up of striped type of muscles. Two types of sphincter muscles are present, that is internal urethral sphincter and the external urethral sphincters. These are the sphincter muscles. These are present to control the storage of urine, and as and when it is required, it is passed out, thrown out of the body. What is the function of this urethra? It carries urine and semen. In case of human beings, it is considered to be urinogenital duct. Whereas in case of females, it carries urine. It is passed out of the body, thrown out of the body. So this is all about the different parts involved in the excretory system of human beings. Now the structure of nephron. Now what is nephron? We will talk now. This is the structure, and it is the basic structural and functional unit of kidney. This is the basic structural and functional unit of kidney. So remember, for MCQ or one sentence answer, this question can be asked. That is, nephron is the basic structural and functional unit of kidney. Now we will study the details about this structure of nephron. Now, where this nephron shows two important parts. That is, this part which is called as Malpighian corpuscle, and what remaining all tubular structures, that is renal tubules. Okay, first remember, this is what the structure of nephron, which is thin walled, coiled, lined by single layer of epithelial tissue, and there are around 1 to 1.2 million nephrons in each kidney. In detail, I will explain you the structure. But basically, this you remember that nephron is made up of Malpighian corpuscle and renal tubule, as I show you in the diagram. Then Malpighian corpuscle, that is made up of Bowman's capsule and glomerulus, whereas renal tubules show all these parts, that means proximal convoluted tubule, loop of Henle, distal convoluted tubule, and collecting duct. If you see the structure, I will explain you again. We'll first talk about the first part, that is the Malpighian corpuscle. What is that Malpighian corpuscle? Whatever is shown here by this blue colored part, this entire structure is called as Malpighian corpuscle. And whatever the tubular structure, remaining tubular structure that is shown here, that is what called as renal tubule. Okay, so first we'll talk about the Malpighian corpuscle. This Malpighian corpuscle is made up of glomerulus and Bowman's capsule. Bowman's capsule is nothing but this cup-shaped structure which you can see, that is called as Bowman's capsule. And whatever these red colored blood vessels you can see in that is a network of blood capillaries, blood vessels, that is called as glomerulus. So Malpighian corpuscle is made up of cup-shaped Bowman's capsule, which shows the presence of a network of blood capillaries in it, that is called as glomerulus. So the blind end of tubule forms a double-walled cup called Bowman's capsule, which surrounds the glomerulus. That is, that cup-shaped structure is having the blind ends of tubules. That is a tubule, but that is considered to be without any end. That is called, it's not open, so that is called as blind end. Now, when you see the internal structure in detail of this Bowman's capsule, how it looks, that is double-walled, cup-like blind end of renal tubule. This is renal tubule only. That is a part of renal tubule only, but that part is not open. So that's why this is called as blind end. And this is cup shaped. So this is cup shaped. Now, what are these parts now? That is made up of two layers. Two walls, outer wall, which is made up of parietal. This is called as parietal layer. Outer layer is called as parietal layer, and that is made up of squamous epithelium, and the inner layer of Bowman's capsule, which is shown here, that shows the presence of special type of cells called as podocytes. These structures which are little bit folded one, right? That is what called as, these are the podocytes. These are specialized cells of this. So inner layer is having the presence, or it shows the presence of podocytes, special cells, whereas there is a space between this inner layer and outer layer. This is the outer layer, that is made up of squamous epithelium, and that is called as parietal layer, and inner layer that shows the presence of specialized cells called as podocytes. And there is a space between the outer and inner layer, which is shown by this part, yellow colored, that is called as space, that is capsular space, or this is also called as Bowman's space, or this is also called as capsule or capsular space, which is present between the outer parietal layer and inner visceral layer. Inner visceral layer that shows the presence of specialized cells called as podocytes. And this Bowman's capsule, where it starts with this tube, renal tubule, this point is called as neck, which is little bit narrow, and then it becomes broader, and then it enters the next part of the renal tubule, that is called as proximal convoluted tubule. That part we will discuss. So is it clear? That is about the first part, that is Malpighian body, which is made up of Bowman's capsule and glomerulus. This is the structure of Bowman's capsule. So the inner wall of Bowman's capsule has certain unique cells called podocytes, which wrap around each capillary. Now, inside the Bowman's capsule, whatever the blood capillaries are present, these are wrapped by these specialized cells called as podocytes. So, inner layer is visceral layer, which shows the presence of podocytes, specialized cells. So, this is again, you can see the structure, that is the outer part is called as Bowman's capsule, which is made up of outer layer, squamous epithelial parietal layer, inner layer that is made up of, that is called as visceral layer, showing the presence of podocytes, and all these blood capillaries are wrapped by these are called as glomerulus. These are wrapped by these podocytes. And now one more thing you can see, the blood vessel, that is the, which is entering arteriole or artery. We are using the word arteriole, which is smaller than artery. So this is what entering, and this is which is coming out. So if you see the diameter of the arteriole, which is entering in the Bowman's capsule and which is coming out of the Bowman's capsule, if you see the diameter, there is difference. That diameter of entering arteriole, that is larger than the one which is going out of the Bowman's capsule. So this is what you need to remember, which is going to help in the ultrafiltration. What is that? We will see now. See, as I told you, which is entering inside the Bowman's capsule, that is called as afferent renal arteriole, and which is coming out of the Bowman's capsule. These are shown by the arrows also. You can remember, these are shown, which is taking entry in the Bowman's capsule, that is called as afferent renal arteriole, which is coming out of the Bowman's capsule, that is called as efferent renal arteriole. And this Bowman's capsule encloses a tuft of capillaries called as glomerulus. These are all glomerulus formed by the afferent renal arteriole, a fine branch of renal arteries. So these are the this is the part of glomerulus, and this is the diameter which is larger of afferent renal arteriole than the efferent renal arteriole. Diameter of afferent renal arteriole is larger than the efferent. So why is it so? I will tell you when we will start the filtration part. So blood from the glomerulus is carried by efferent renal arteriole to a lesser diameter, as I told you just now. Now this is the next part of the nephron, that is renal tubule. Now I explain this part, that is the Bowman's Malpighian body, that is cup-shaped Bowman's capsule, and which wraps which surrounds the glomerulus, and this remaining part, now this which now starts this part of the Bowman's capsule, this part is called as neck, and then this neck shows the foldings, so many foldings near the Bowman's capsule are seen, and that part which is near to the Bowman's capsule, this is called as proximal convoluted tubule. Then which you can see the hair pin like structure, U-shaped structure, that is called as, here this loop is called as loop of Henle. Then which enters here the distal convoluted, but here again this tubular part is folded, this is called as distal convoluted tubule, and then at the end it enters the common duct called as collecting duct, where urine is collected and passed out, taken to the ureter, through ureter to the urinary bladder. So this is the collecting duct. So this is the entire structure of renal tubule, which includes 4 parts. The part which is folded and nearest to the Bowman's capsule is called as proximal tubule or proximal convoluted tubule. These are folded, so convoluted. Then it enters the descending and ascending loops of Henle. First, this is U-shaped structure, this proximal convoluted tubule, and then it continues in the loop of Henle, which is the U-shaped structure, larger one. This is the loop, so this is called as loop of Henle. This is the descending part of the loop of Henle. This is the ascending part of the loop of Henle. Then it continues in the distal convoluted tubule, where again this tubule is folded, and this region, folded region of this tubule is called as distal convoluted tubule, and then it enters common duct called as collecting duct, where urine is collected. So this is all about the renal tubule. One by one we will now talk about each part of the renal tubule. That is, the first is what? Proximal convoluted tubule. Now when you see, this is the Bowman's capsule. Now this double-layered wall, now it is entering, this is the neck, which is a little bit narrow, and then it becomes broader. This folded structure is called as proximal convoluted tubule, that continues further and forms a next part, that is loop of Henle. Okay, so whatever the renal tubule starts from the Bowman's capsule, this is highly coiled, and that's why called as convoluted tubule. Whereas next part is loop of Henle. Now loop of Henle shows 2 parts. One is ascending limb, one is descending. If you see this convoluted or coiled structure of proximal tube, it continues here. In your diagram also you can see it is clearly shown with this page number whatever 181. 15.7 nephron and peritubular capillary network. You can see the diagram. Try to understand this diagram. I'm explaining it here. So this part which continues from the Bowman's capsule and which is folded is nearest to the Bowman's capsule, that is proximal convoluted tubule. Then it enters this U-shaped structure loop, that is called as loop of Henle, which shows 2 parts. One is descending, that is called as descending loop of Henle, and that looks like a hairpin U-turn structure, hairpin turn structure, and the one which is ascending, that is called as ascending limb of Henle, and then it continues to form the next convoluted part, and that is this distal convoluted tubule. Now one thing you remember in this diagram, when this proximal tubule enters the loop of Henle, the part of loop of Henle of the descending limb, this is narrow, whereas the ascending limb here, this is ascending limb, that is thick. You can remember the proximal part of ascending limb is thin, whereas the next part which is growing, that is distal part, that is thick. This is the descending limb and this is the ascending limb of loop of Henle, which is shown by this yellow colored arrow. Okay, try to understand. This arrow is showing the loop, ascending loop of Henle, where this part of ascending loop is thin and the upper part, that is the distal part, is thick. Now there is also one reason behind it, where it helps in reabsorption as well as filtration, secretion, filtration, reabsorption. This is very helpful. So this loop of Henle continues into again the convoluted or folded part, and that is called as distal convoluted tubule. See, this is the ascending limb of loop of Henle, which is again here little convoluted part it is shown, that is the folded part, that is called as distal convoluted tubule, and the thick ascending limb that it continues is called as distal convoluted tubule. Now when this distal convoluted tubule, it continues into collecting duct. Some initial collecting ducts unite to form a straight collecting duct which passes through the medullary pyramid, and this tubule, distal convoluted tubule, which is as the initial collecting duct is present in the cortical region. That part I will show you in the next diagram. So remember all the parts of the renal tubule. So blood supply of nephron, how it takes place? From here, the efferent arteriole emerging from the glomerular forms fine capillaries networks, that is into the peritubular capillaries around the renal tubule. Now what are peritubular capillaries? These are all renal tubules which are surrounded by the blood vessels. So these are all called as peritubular capillaries. So this is the network of peritubular capillaries. This is the main part, that is the nephron showing the Malpighian body, that is Bowman's capsule and glomerulus, and then renal tubules, and renal tubules are surrounded by the network of capillaries. These are called as peritubular capillaries. So efferent arteriole and afferent arteriole, if you see, the diameter is different, as I told you, that helps in the ultrafiltration. So this is what the structure of the entire nephron, where you need to remember the structure of nephron in the form of Malpighian corpuscle and the renal tubule. And the renal tubules are covered by the network of capillaries called as peritubular capillaries. And here loop of Henle, which is covered by these nephrons or the peritubular capillaries, is called as vasa recta. This you remember these terms. So vasa recta is highly reduced or absent in cortical nephrons, but it is well developed in juxta medullary nephron. What is that? I will tell you in the next part. But I will stop here, where I will revise a little part of this, where just have a look in the modes of excretion: ammonotelism, ureotelism, and uricotelism. Okay, so these three modes of excretion, the human excretory system involves 4 parts, that is a pair of kidneys, pair of ureters, urinary bladder, and urethra. So we studied the structure in detail, that is how it is, its color, shape, location, weight, length, breadth, etc. Isn't it? About the and weight, etc. Then we studied the internal structure of kidney also, where 3 parts: outermost cortical, middle medullary, and innermost as pelvis. So this is the inner part of, or the longitudinal or vertical section of kidney, where these 3 regions, they show the presence of renal pyramids and even columns of Bertini. And the process of micturition, then ureter, the next part, then urinary bladder, then urethra, the structure of urethra, and at the end we studied the structure of nephron, which is considered the basic structural and functional unit, which shows two main parts as Malpighian corpuscle and renal tubule. Malpighian corpuscle includes Bowman's capsule and glomerulus, whereas renal tubule includes proximal convoluted tubule, that is called a PCT, loop of Henle, U-tube structure or U-shaped structure with the two descending and ascending limbs, and distal convoluted tubule, DCT, and the last part collecting duct. So this is all about the structure of nephron. Now I will stop here